subRosa shapes its method and means a lot in order to reach the audience more effectively. Mostly we do it by what we call situational work. We really work with a specific site and a specific situation. And we do performance art that is not theatrical or narrative and often takes place not in an art venue. Sometimes the audience will be passers-by who aren't expecting to be encountering or engaging with art. And that can really open up a lot of interesting dialogue. For example, in our performances that address the value of egg donation, some students are really interested or upset to learn the actual value of their eggs on the market, or they're upset to realize that it actually is a market. And other students are really interested because they discover that they have a commodity that they didn't realize they had before. Sometimes we've gotten into pretty heated debates with people who, in their family, there's congenital defects that they might like to cure through some of the reproductive technologies. When we do our performance about organ donation, that's a really emotional topic for a lot of people. And so we're not trying to tell people not to engage in these technologies, we're just trying to encourage some discussion and debate about some of the ways that the economies of it work. So people who have already some sort of economic or health relationship to the topic get really engaged in the issues. In some ways, I don't think subRosa worries about being considered viable as artists. I mean, of course we do, but that's not why we are artists. Like, we're not artists in order to be viable artists. We're artists to produce work that we feel is important. I think that the arts are always in a state of evolve or perish. I think art is usually responsive to the economies and conditions of the times, no matter when it happens. So I think it's sort of a continuing evolution or perishment. Maybe when you're looking back in history, it sort of starts to blur together in some monolith, but I don't think that's actually how it happens. I don't think that necessarily pumping more funding in would change the way that art is produced. It would probably change distribution and production standards, but really what needs to change is the way that, you know, this culture, which is actually like kind of a complex thing, not a monolithic one, but if this culture changed the way that we thought about art and wanted to receive arts, that would change it more than funding would at the root. It's not the money, it's more like the respect and the ability to kind of read art that would maybe change things. I also think that going back to the sort of divisions between art and design and business and the academy, I think that it could be interesting if it was not so taboo for serious artists to also be able to engage in kind of cottage industry models where it wasn't so taboo to sort of blend the different ways that you make a living. So, for example, it becomes really complex to get arts grants if you also do commercial work, and that's too bad because it kind of is a double bind for a lot of people who need to make a living. subRosa is a collaborative practice made up of different individuals, and we each have our own set of influences. We're also an intergenerational collective, so we're really influenced by different moments of history and the different places on the earth that we've lived. One thing that the members of subRosa have in common is that we draw influences from a really wide range of people, moments, disciplines. Definitely different forms of activism, civil rights in the US, human rights globally, and the feminist movement of the United States from the late 60s on have been really big influences on our work. Most of us read a lot of different kinds of writing, from theory, political science, literature. I can only really speak personally for myself. I'm I'm pretty influenced by some of the Fluxus movements and contemporary neo-Dada, as well as identity-based artworks and post-minimal process work from the 70s. It's kind of a boring answer, but I think it's more interesting to say most of the members of subRosa draw really widely from a lot of different influences, cultural as well as artistic and political.
But exchanging DNA is not reproduction necessarily. It's just, however, when the cell that has exchanged DNA reproduces, then you hope that it's going to reproduce that new DNA that it got. That's what we're hoping our E. coli is going to do now. It's going to reproduce and be resistant, So that's kind of, we're trying to understand it ourselves by doing the actual experiment, because then you can, I think, understand it much better. And we may fail completely. We may have done it completely wrong. It's the first time we've ever done it, Did it without, any kind of scientific instruction, because I think it's really, it's sort of like do it yourself, kind of learn it yourself. And of course, this is an experiment that's been done a million times. It was done early in the 70s. It's the classic recombinant DNA experiment. So we're learning it, because it'll be a basis for doing other things.
This is what's called streaking a plate with bacteria.
Isn't that dangerous to touch it?
No. I'm not touching the bacteria, I'm touching the plate. But this is actually a standard lab experiment that a lot of high schools do in their biology labs. And it really follows all of the principles of cell culture.
This is a sealed, store-bought carton of organic yogurt, right? This is going to be my starter. And it says on it that there's 6 live cultures in here.
No, 5,000 years ago minimum is when we were making yogurt as a human civilization. Part of the same process that she was doing over there is going to be done over here. It's what we call the scientific method.
They also, but anyway, it kind of digestion enzyme, and that allows it to sort of eat its way. Inner cells start to cleave and split apart, and those cleavages can be separated and they can become whole new organisms. They can also be, because they're pluripotent, they have great value because they can also be teased and turn into basically any cell in the body, whether it's lungs, liver. So those cells are very valuable, especially in the early stages of cleavage.
We splice it in.
Splice it in.
That's the
recombinant.
The topic of this experiment is gene splicing. We're taking 2 proteins and adding them to the genetic material of the E. coli, and now magically the new bacteria that we will grow on the petri dishes, will be resistant to the antibiotic.
Streaking is a process by which we spread cells over the surface of a nutrient medium in order to let them grow.
This is a plate that we streaked with E. coli bacteria. You can see a lot of growth here, right? You can see this white stuff. That's what's basically in your guts, right? And it was streaked on a plate that had no antibiotics in it.
Sort of rub this back and forth like very lightly. Like that. You just touch the surface. It's very much like jelly, and you don't want to break it. See there, I kind of accidentally dug into it. You don't want to dig in, you just want to evenly distribute it on top of the jelly. Go back and forth that way.
Really feel like you're spreading like a very soft little,
Vectors is a DNA molecule that can be used to carry foreign DNA fragments. In other words, these are things that we can change. We can change the DNA of these little vectors and put them into the cell. Yeah, there's a lot of them. And, you know, and they're little bits of DNA that are floating around in the E. coli. See, bacteria is really an interesting organism because bacteria exchanges DNA with other bacteria all the time. You know, so scientists have figured that out, and so they're just kind of in a way copying, you know, in the lab and directing in the lab what bacteria naturally does.
E. coli and its metabolic processes in our gut helps our body make that vitamin so that we can have healthy blood clotting go on. If this person has a cold or something and is taking antibiotics, the antibiotics go through the whole system and they're going to kill the normal flora. And so that's why we want resistant bacteria. That's why we could live with resistant bacteria. We live without it, and yet if we do take that antibiotic and it kills a lot of our E. coli, what happens then is something we all know and don't love, diarrhea.
Yeah,
Plasmids, which are basically little process happening. I want to learn about the process because it's like, yeah, because it's a really basic process of biotechnology. You know, we're trying to learn this experiment so that we can understand it better. But basically, the plasmid has actually areas on it that are called cloning sites that can be broken open.
As a scientific group is what was actually done the first time in 1974 by 3 scientists in the lab working with E. coli, and they